So, the molecules that are present in bacterial growth media are very often what controls the expression of the bacterial genes. And this makes a lot of sense because gene expression is a process where the cell determines which machinery it wants to get out for the situation that it happens to be in. So, maybe if a bacterial cell is in glucose media, it will get out the glucose machinery, versus if it's in lactose media, it will get out the lactose machinery. But these are just the well-studied examples, and the molecules that influence gene expression don't have to be energy sources, so that's why we're going to refer to them very generically as molecules. And these molecules, through a sequence of events, signal specific proteins called regulatory proteins to regulate the actions of other proteins involved in gene expression. So by now you should know that RNA polymerase is the protein that's responsible for transcribing DNA into RNA that is then translated into proteins that can go and perform functions around the cell. Such as a regulatory protein, but proteins don't have to be regulatory. But in this video, we're going to stick to regulatory proteins. So anyway, what the scientific community has done is name these regulatory proteins in a very generalized way according to how they affect the other DNA binding proteins that they come into contact with. And the names are fairly straightforward. They're either activators or repressors. And I think you can guess pretty easily that activators increase the transcription of the gene that they're regulating, whereas repressors decrease the transcription of the gene that they're regulating. So, as you may have heard, there are many steps to genetic expression and transcription, and different regulators may regulate genetic expression at different steps along the way. However, the vast majority of regulators act during what's called transcription initiation, and this makes a lot of sense in terms of energy efficiency, because by deciding whether or not to express a gene before the process has even begun, the cell prevents itself from wasting energy producing a protein that it doesn't need. And then the cell doesn't need to worry about having something running around finding every mRNA that was transcribed by accident or every protein that was translated by mistake and having to deal with them after the fact. It can just deal with them during initiation. But post-initiation regulation does occur, so why would we want that? Well, sometimes a specific amount of expression or an expression speed is needed for the cell to perform its functions normally. And the cell sometimes best achieves that desired speed or rate by having multiple points of influence across the transcription process. And then in that way, it can better define exactly the rate that it needs for this protein to be constructed.